Hello lovelies, in this video we're going to be looking at stem cells for your A-level biology. So how stem cells are made, how they could be used, and then thinking about the implications for wider society.
Stem cells and differentiation. Multicellular eukaryotic organisms start off as a single, undifferentiated cell. These cells divide by mitosis to form a ball of stem cells. So we all started off life as a zygote, which was one fertilized embryo, and then our cells begin to divide by mitosis, and we call this like embryonic cell division, and that's how the embryo begins to grow. And these cells here are undifferentiated, and they are known as stem cells. Stem cells are undifferentiated because all the genes in the genome of each cell can be expressed. The process of differentiation happens as different genes begin to be expressed or not expressed. So some genes will be switched on and some genes will be switched off in the cells. And that's how they begin to form specialized cells which are adapted to carry out a specific function. So these stem cells all start off the same, but as they begin to differentiate, they can become bone cells, red blood cells, nerve cells, epithelial cells, neutrophils or phagocytes, so white blood cells, or muscle cells, for example. And you can see that all of these cells are very different from each other. Through the process of differentiation, there are some changes that happen to the cells in order for them to end up being a specialized cell. So often the cell shape changes. You can see that mostly here with the nerve cell, for example. The cell contents might change, and the proportion of organelles changes. So how many mitochondria they have, how many ribosomes they have. That is what changes in order to allow that cell to carry out its new specific function. Okay, so on to pluripotent stem cells. These are cells that can differentiate into any of the three germ layers. So we have ectoderm, endoderm, and mesoderm. This is what I was talking about by the three kind of pathways. So the ectoderm, if you're a pluripotent stem cell that can form ectoderm tissues, then you're going to be able to form all the things like the nervous system, the skin, sort of our outer layers, the things that kind of start forming first. Then you've got your mesoderm normally, which goes in the middle. So that's kind of the bones, the muscles, the connective tissue. And then the endoderm is kind of like your innermost layer, and it forms the internal organs. So your pancreas, your liver, all of those things. So any tissue in the body or any plant tissue can be made of these three, but they will be three different types of pluripotent stem cells. They'll either be ectoderm, endoderm, or mesoderm, depending on where they are in that ball of cells. So you can see we've got layers forming. So cells from the inner mass of a blastocyst, which is 4 to 7 days, are pluripotent because they've started to differentiate into these three layers. So really we're looking here, and it would be kind of cells in these layers in here, not necessarily the ones on the outer layer, but the ones that have started to organise themselves in the middle. They will be pluripotent, and they can obviously become these three. Plant stem cells are also pluripotent, and they're found in the cambium. So if we look at, so this is one of those, if you cut through the stem of various plants, you'll see the vascular bundles, which is obviously the xylem and the phloem, arranged in these little wedge shapes around inside the stem. So this is one of those little wedge shapes, and we have the xylem, which is always on the inside, and we have the phloem, which is on the outside. And then in between, there is this layer. It's not super easy to see in this one, but it's what we call the cambium tissue. And that's where our stem cells are that are able to keep producing xylem and phloem and other tissues and sort of growing the shoot tip or the root tip and extending it. And that cambium is present for the entire life of the plant. So that blastocyst is there for maybe a few days, and then it's gone, and we get into more differentiated specialized cells that lose this ability. Whereas in the cambium tissue, these pluripotent stem cells are there for the entire life of the plant, which is how you can chop a piece of plant off, take a cutting, and that stem where you've cut it clean will start being able to produce and grow roots because it has those stem cells there, and you can form and clone a new plant. You can't do that with most animals because chopping a part of them off will not leave any stem cells that are able to then regenerate into all the different tissues needed. So multipotent stem cells, these are now, we're getting into adult stem cells. Obviously the previous two were found in embryos, so they are embryonic stem cells. This is now our first example of an adult stem cell. These can divide from different cell types, but in a limited range, and it obviously depends where the stem cells are found in the body, what they can do. So stem cells from bone marrow are a good example. They are multipotent stem cells. Some genes have been switched off, but they have the genes from different cells still available and that could be expressed. And so they're able to kind of go down this pathway of differentiation, and the cells in the red bone marrow go on to produce immune system cells, platelets, and red blood cells. Whereas if we look at the image, we can see that there's also yellow stem cells or yellow bone marrow present, and that goes on to become cartilage, fat, or bone cells. So that's how the bone can regenerate itself, and obviously it's where we can regenerate cartilage, which bone requires, and fat cells as well. Okay, so like in GCSE, we still need to be able to talk about the differences between adult and embryonic stem cells. So the source for most embryonic stem cells is unused IVF embryos, because these will be donated for research if they're not going to be used by couples who have gone through the IVF process and put some embryos in the deep freeze. Then if they've had maybe 1 or 2 children, or it's not worked, or they don't want any more children, they don't want to use any more of those embryos, they can be donated for stem cell research instead of being destroyed. Whereas with adult stem cells, as we've seen, bone marrow is one of the easiest places to get these stem cells from. It can be removed with a simple operation that carries little risk, because you don't have to be completely unconscious or anaesthetised, but it can be quite uncomfortable and painful afterwards for the person that is having the operation. So what are the uses of embryonic stem cells? Well, they've been used to grow new organs or tissues for treatment for a variety of conditions. For example, anything to do with the nervous system, so Parkinson's disease or spinal cord injuries, being able to develop new nerve tissue and new nerve cells and put them back into patients suffering with this, where those cells have been damaged or are being degenerated, and then they can help them improve their function. They could obviously improve the quality of life for many people, and also reduce the need for organ donation. If we could grow new organs from them, then we don't need to find donors in order to get them. Bone marrow is already being used and has been used for a long time, so bone marrow transplant, being able to donate your bone marrow, so your stem cells, to someone else, is normally used to treat blood conditions where abnormal blood cells are the issue, so things like leukemia, lymphoma, sickle cell disease, and SCID. They have also been used to treat paralysis, because spinal cord injuries can be used to replace nerve tissue, so they can be used as well as embryonic stem cells. The best thing about the adult stem cells partly is that they remove the issue of rejection. So if you get donated embryos from somebody else, they will not be genetically the same as you. But if you have your own bone marrow cell samples, then they are going to be genetically the same as you. And so it's less chance that your body's going to obviously reject it. Issues with embryonic stem cells, again, this is very similar to what we looked at GCSE. Some people are objecting to the use of embryonic stem cells because they see that it could be a potential fetus. If it was implanted into a womb, it would grow into a new fetus and it could become a new human. Some people believe that because fertilization has happened, that the embryo has a right to life and it isn't fair to be used. But obviously these embryos would have been destroyed if the people no longer want them or need them and are no longer willing to pay for them to be stored in a freezer. They would just be destroyed. They're not likely to be implanted into a womb. But this is some people's issue with embryonic stem cells. Obviously also as well, the kind of storage is expensive and can be expensive to store those over a long period of time. Some people, babies now being born, if there is a potential for genetic disease or a high risk of disease in the family, they're suggesting that storing the umbilical cord, blood or placental tissue could be stored, because it could be a source of stem cells that are genetically identical to that baby that's been born, and store them in case they need stem cell treatment later on in life. And obviously that can be quite expensive, and that is really only available to people who can afford to do that, which is somewhat controversial. The issues with the adult stem cells is that they're only multipotent. They are not totipotent or pluripotent. So they cannot differentiate into as many cell types as embryonic stem cells can, which means they can't help as many diseases or be used to create as many tissue types as embryonic stem cells can. So there's a limit there. There is a potential way we can solve that problem of the adult stem cells not being able to be plastic or potent enough as the embryonic stem cells. There is new ways of creating what we call induced pluripotent stem cells. So these stem cells are created in a lab, They're taken from specialised cells, so the easiest one normally to take is skin cells because they're easily accessible and it's not very harmful to take them from a patient. And then these cells are reprogrammed to become pluripotent. So these are not stem cells, they're just normal skin cells that would not have the ability to do anything or become any other type of cell. They are fixed, they're differentiated, they are specialised. But we can reprogram them using maybe a viral vector, which can introduce transcription factors. Again, we're going to look at transcription factors in more detail in the next video. But these are just proteins that can control the expression of genes. So if you can insert these proteins or insert DNA that's going to change or trigger this change in the cells, it will cause them to turn back on or switch on all of those genes that are normally expressed in a pluripotent stem cell. So that's like an embryonic stem cell. And so we can take those now induced stem cells and then we can use them as we would embryonic stem cells. But we've got these from a patient, and so it is genetically the same as them. So we have the benefit of both worlds. We have the pluripotency of an embryonic stem cell, so the ability to turn it into many different cell types, and we have the genetic makeup that makes it identical to the patient, so it removes that chance of rejection. And that's why these stem cells are so useful and interesting and very important in terms of the way we can move forward with stem cell research. So clinical trials are already undergoing to look into the use of these induced pluripotent stem cells to do things like replace diseased or damaged organs or to replace damaged tissue. So we can use these new cells to treat loads of different things. So neural cells, neurons, nervous tissue, all of that we can use to treat neurodegenerative disorders. So things where the nervous tissue is being attacked, or like we said, paralysis, so spinal injuries, Parkinson's, even potentially Alzheimer's, things like that. Skin cells can be grown. So instead of having to get a skin graft, so instead of having to take skin from elsewhere on the body or getting skin cells from a donor, you could create a skin graft to replace burnt tissue. So if you suffered really bad burns, then you could grow new skin to replace and cover that burnt skin cells. Pancreatic cells, literally growing a new pancreas or growing new islets of Langerhans that will be able to produce insulin to help treat type 1 diabetes. There are lots of potentials here for this treatment. So that is stem cells, and this was just kind of an introduction to the idea that genes can be turned on and off, and that turning on and off of genes or increasing or decreasing the expression of genes controls what type of cell a cell becomes or controls its specialization. So next video we're going to look at transcription factors, as I said, which is kind of the actual biochemistry of how the expression is changed. And then we're going to look at the next level of that, which is epigenetics. And that's another way that gene expression can be affected. And we're going to kind of tie this together with sort of looking at mutation and looking at gene expression and think about how various things can trigger or cause cancer, because cancer is a way that cells change and the structure and function of the cells change dramatically in order for it to become a cancer cell. So that is another type of how gene expression can be changed in order to change the way a cell works. Stem cells in medicine.
So when we talk about stem cells, we're mostly talking about pluripotent stem cells that can differentiate into any type of cell. Obviously, adult stem cells are slightly less effective because they are only multipotent. But we've got all these different sources. So embryos we've looked at, umbilical cord blood we didn't look at, but that is similar because obviously the umbilical cord is formed from that early embryo. Adult stem cells, as we've said, blood, brain, muscle, bone, skin, there's a few examples of adult stem cells. And then IPS, or the induced pluripotent stem cells that we can produce in the lab from normal cells, and then we induce them, as we said. So we need to know these, but we also need to think about how we can use stem cells and why they're useful to us in different forms of medicine.
What can we use stem cells for? Some of them can be used for a bone marrow transplant. So after cancer treatment, a transplant of bone marrow will help a patient recover after their white blood cells and other blood cells have been damaged. Growing tissues using stem cells that then can be used to test drugs is a better way of viewing it than testing on animal tissues, because these cells will be human. Observing stem cells and how they grow and develop can help scientists to understand how cells function and how they grow and develop inside humans.
They can be used to repair or replace damaged tissues. For example, we could grow a new pancreas or new pancreas beta cells to help people with type 1 diabetes who can't produce insulin. In the future, we could look at actually using iPS stem cells, programming them in order to be able to grow a new organ for a transplant for a patient. So take some of their own cells, program them to become pluripotent stem cells, and then use those stem cells to grow new organs or tissues that could then be transplanted back into the patient. This is really important because if we're using the patient's own cells, then no immunosuppressant drugs will be needed to be taken for the rest of their life.
Ouch! This is why in some videos I explain scratches.